Hi. So in our last discussion, we looked at the Oparin-Haldane hypothesis, which gave a series of steps by which simple non-living molecules under the prebiotic conditions could have given rise to life. Now, as I said, this is one of the biggest and most fundamental questions in whole of biology, and therefore people have spent a lot of time and effort to validate each component of those steps, and countless experiments have happened over the last 100 years or so. So we do not really have time to go into it in great detail. I mean, it can easily be taught as a full one semester course or maybe more than that. So what we are going to do is have a quick bird's eye view of some of the key insights and some of the key experiments that has shaped our understanding of how life originated. So the first step, if you remember, is that under the conditions of primitive earth, organic monomers, what do I mean by that? Amino acids, nucleotides, sugars, etc. can really be formed. Now, what are the conditions of primitive earth that we are talking about? Now, we obviously do not have any direct evidence of what those conditions were, but we have some circumstantial evidences from, for example, what happens when there is a volcanic eruption, what kind of gases come out from that. The other major thing that we have is that if these molecules, they must have formed in the open, okay, directly in the contact of atmosphere, because at that point you do not really have membrane organelles. So under those conditions, if the condition had, or if the atmosphere had as much oxygen as we have today, then there were good chances that these reactions, these monomers, they would not even have been formed. How do we know that? Because of the properties of those monomers. Therefore, it is generally thought that the early atmosphere was reducing and obviously had much less oxygen, and more importantly the source of energy was different. So today, for living molecules, the primary source of energy is coming through photosynthesis or ATP hydrolysis and so on. But at that point of time, these molecules are not there, these pathways are not there. Therefore, the primary source of energy will be in the form of electric discharges that are happening in the atmosphere, lightning, etc. during thunderstorms, and heat. Heat from where? Remember, Earth is still a young planet, so it is still very hot. So a lot of heat is directly coming from the surface. Now, as far as the electric discharge and heat is concerned, people are reasonably confident that that is how things were. In terms of the reducing property of the atmosphere, there is some disagreement amongst the scientists about whether it was strongly reducing or whether it was weakly reducing or whatever. So different people have said different things, but it is generally understood that the level of oxygen was definitely less than what it was today. So main question, can organic monomers be formed under such a situation? And a very famous experiment that first tackled this question and showed it rather nicely that it can happen was this one, due to Stanley Miller and Harold Urey. So although this is known as the Miller-Urey experiment, it is generally acknowledged that the idea and the entire execution was due to Stan Miller, who at that point of time was a PhD student with Harold Urey. Urey was already a Nobel laureate and extremely famous, but this was entirely Miller's game, and frankly Urey more or less has recognized it throughout his life that it was Miller who did all the job. And in fact, if you look at the paper, I am showing you the reference over here, you can see that the paper only carries Miller's name, but somehow this is known as the Miller-Urey experiment. So what exactly was done? So Miller was trying to simulate the prebiotic conditions. So you have two major components, the ocean and the atmosphere. So in order to simulate the ocean, what he does is he takes a glass flask and puts some water in it, and then he heats it using a heat source. What happens? This will lead the water to become vapor, right? That is what happens even today. You have sun rays falling and water turns into vapor. Then this vapor, this goes up, and in the context of the setup, this goes up through this tube, and then it enters the atmospheric component over here. What is the atmospheric component? It is another big glass flask, and in that flask he put things like ammonia and methane and hydrogen. Now, why did he put these chemicals? Because this was his best guess at that point of time in 1952 about what the atmospheric composition would have been like. Then, in order to simulate lightnings, he puts two electrodes, and using an electric connection basically creates a continuous spark between those two electrodes. So, as a result of this high source of energy, these gases plus water, there is reaction that is happening in this simulation of the atmospheric component, and then this stuff, this condenses, and then it comes down over here. So, here he puts a condenser to cool the whole thing, and this is analogous to the rainfall essentially. And then this comes over here, comes down, and he puts a trap over here, so that from here he can take samples and analyze them. There is one more trap over here to understand what is happening in the oceanic component, this part, and then he sets this up, then just lets this run for 1 week. Okay. So, I have a very nice video which shows you quickly the same stuff 3D animated, just to show you how the components rotated, went from one part of the setup to the other. So, I will just try to play this. Yeah. Right. So, that blue arrow, that is showing you the path of the water vapor actually. So, this is, as I said, the oceanic component. So, this is being heated, and then you have the formation of water vapor as shown by these bubbles, and this water vapor rises, and then through this tube it reaches the simulation of the atmosphere. So, in this component here you have the electrodes which are continuously sparking. Because of that, chemical reaction happens, condenser cools the whole thing down, and then this again comes back over here. So, there is a continuous circulation that is happening, and as this says, at the end of 1 week, as much as 10 to 15% of the carbon was now in the form of organic compounds. And yeah, so these are the people who made that excellent video, and the results from this experiment were absolutely astounding. So, after a week, the solution had turned deep red and turbid. Remember, this is 1952, so analytical techniques are still not as good as they are today. So, at that point Miller had to do paper chromatography to figure out what are the various things that are there, and he identifies 5 amino acids, proving that the monomers of proteins can be formed from inorganic and organic and pre-life molecules. So, it does not end here. So, this is happening in 1952. So, somewhere around 2007 Miller retires, and when he retires, turns out that there were several samples that had been created from experiments done during that time, not the 52 one, later experiments, and these samples had actually been sealed by Miller and kept. So, when he retires, this goes to the group of I think his name was Jeff Bada, and Bada then takes these things and ends up reanalyzing these samples. But since he is doing it in 2007, he uses more modern techniques like high performance liquid chromatography and time of flight mass spectroscopy. And when he does that, he figures out that the same samples actually contained 23 amino acid and 4 other amides. Okay. So, the yield was actually much greater and more diverse than what Miller had actually ended up identifying from his primitive technique. So, this was a massive thing in the history of origin of life research, and this conclusively proved that the fundamental building blocks of life could indeed arise from non-living matter. Now, this was primarily about amines and amino acids. What about the other kinds of molecules? Okay, you need so much to create life. So, his research over the last 100 years has shown that almost all the biological monomers that are needed for life, all of them could have been formed on the prebiotic earth. So, here is a video, I am sorry, review that I am giving you the reference for, which essentially talks about the various molecules and under what conditions they were synthesized. Very, very comprehensive review. So, now that you have the monomers, but remember all this is happening in the ocean. The ocean is huge. So, the concentration of these compounds, even if they are being formed, is going to be very, very low. And even if a lot of time passes, they are still not going to become too high. And if you have to go from monomer to polymer, at some level or at some point the concentration needs to become high, otherwise polymerization reactions do not take place. Okay. So, how will that concentration happen, or how did that concentration happen? So, in order for the monomers to come together to become polymers, you need some mechanism by which they will come together. So, turns out that there are multiple possible ways in which this could have happened, and I am just talking about 3 of such possible solutions. There are many more. So, by the way, when I am talking about possible solutions etc., these are not mere hypotheses. These have been explicitly shown by people to work in order to create polymers from monomers. So, for example, there are many kinds of clays, and the most well studied example is this thing called Montmorillonite. I hope I am pronouncing it right. So, if you have clay, then what happens are 2 or 3 things. First of all, the clay, it's a adsorbing material. So, it can absorb various kinds of monomers on top of it, which obviously increases the concentration. The second thing that happens is that many of these clays, they act as catalysts, and in particular they can catalyze the phosphodiester bond, which is a very important thing in the formation of polynucleotides, DNA and RNA precursors. Okay. So, that is one thing that people have nicely shown again and again, that clay is one of the major ways in which many monomers can be concentrated to become polymers, or rather many monomers can polymerize, basically forget concentration, they can actually polymerize. The second huge candidate that people have are the hydrothermal vents. Okay, so you know that these are vents typically at the bottom of the ocean, where continuously heat is coming out, and the chemistry near these hydrothermal vents is absolutely fascinating. Okay, I am not an expert on it, but when I look at it, I can see that there is a lot of complicated stuff going over there. Now, apart from the chemistry, you also have very peculiar structure in these hydrothermal vents, which are kind of like porous kind of structures, wherein people have shown that all these or many of these monomers can actually attach, just like they can attach on the surface of the clay, they can also attach over there and thereby form local high concentrations, which obviously can be good for polymerization. The other thing over there is that the chemistry is such that people have shown empirically and theoretically that chemistry is extremely conducive for many of these polymerizations. That is why many scientists believe that the initial polymerization leading towards origin of life might have happened at these hydrothermal vents. And the third major thing that people have shown is that suppose you have a mud flat, okay, or a tidal pool. So, what happens? Water comes and then it evaporates, but when it evaporates, obviously the concentration increases. Now, after some time it becomes dilute again and then again evaporates. So, if this keeps on happening again and again, then the concentration of the monomers is going to increase over there, which might end up promoting polymerization. Now, please appreciate one thing: A, these are just 3 of the many possibilities. B, nobody is saying that these are mutually exclusive. It is entirely possible that all these things worked simultaneously, and some of the reactions happened in one kind of surface, some of the other reactions happened on another kind of surface. So, this is how one can go from monomers to polymers. But now, I mean, polymers are, well, just molecules, right, but somewhere there has to be function, otherwise life will not work. So, in order to understand how you go from polymers to functions, let us go back to the definition of life, the NASA definition of life that we had discussed in our previous discussion. So, if you remember, the definition was that life is a self-sustaining chemical system which is capable of Darwinian evolution. Now, let us unpack this definition. You have chemical system and you have Darwinian evolution. So, obviously, if you have a chemical system, then that chemical system has to be doing something, it has to be performing some roles, it has to be, there has to be some catalysis or some kind of reactions happening. But more importantly, if you are talking about Darwinian evolution, okay, remember natural selection, and remember I had said that in order for natural selection to happen, you definitely need inheritance of information, right. So, any self-sustaining chemical system which can undergo natural selection will need a chemical structure which will be capable of encoding complex genetic information, and that complex genetic information will need to be transmitted to the next generation. So, there has to be a way of making copies as well. Without this, the whole thing is not going to work. Okay. So, we have three major kinds of molecules here, proteins, rather two major kinds, proteins and nucleic acids. Now, proteins, they do, under normal situation which is present day biological systems, the proteins carry out all the metabolic reactions through catalysis, including that reaction of making copies. So, DNA polymerase is a protein, right. However, the issue with proteins is it cannot store or pass information to the next generation. So, protein does part of the work. What about the nucleic acids? The nucleic acids, as we know, they store information which can be used to make the protein either directly or via the mRNA, but the issue with them is that they cannot catalyze reactions or they cannot copy themselves, right. In order to copy the DNA into RNA or the DNA to DNA, you again need protein. So, these two molecules, these two classes of molecules, they are very, very tightly subfunctionalized, and hence the question is that how exactly are they, or rather which exactly came first, because it is a chicken or egg problem. In fact, many people call it the chicken or egg problem. So, neither can reproduce or function without the help of the other. So, if that be the case, how did the first one evolve? Whichever it is, either DNA or protein, whichever it is, came first. So, turns out that this was a conundrum for some time. Before the conundrum got resolved somewhere in the 1980s, when two people, Thomas Seck and Sidney Altman, they discovered that certain RNA molecules, which they called as ribozymes, apart from carrying information, they can also act as catalysts. Okay. So, this led, or rather this implied, that these two functions, working as a catalyst and working as a information storage and replication molecule, both these functions can actually be in a single molecule, and that single molecule which can do both is the RNA. Now, although this comes as a surprise, if you think about it a little bit more closely, and if you look at what we have understood about RNA biology in the last 20-30 years, it actually is no longer a surprise to modern biologists. Why? So, because nowadays we know that RNAs, they play all kinds of role in our body. So, for example, we talked about siRNA or piRNA or non-coding RNAs and their roles in gene expression and all kinds of other things, right. So, they do play a lot of regulatory roles, and on top of that, if you look at the ribosomes, okay, so the ribosomes are one of the most conserved molecules or things in the whole of life, tree of life, and ribosomal proteins are amongst the few 30-40 proteins which are common across every single known organism. Now, if you look at the ribosomes and if you look at how a ribosome functions, you know what it does? It basically, catalyzes the function, the joining of those amino acids to form the polypeptide, right? The catalytic function, which is doing the joining, that part is actually not done by a protein. So, although ribosome structure contains many proteins, but the real job of the ribosome is actually performed by the ribosomal RNA, okay, which essentially obviously is a ribozyme. So, this straight away tells us that, and given the fact that ribosomes are extremely fundamental, extremely well conserved, this straight away tells us that RNA did have a very important role in catalysis of reactions, today something which we almost exclusively tend to associate with proteins. But now the question is that, okay, so if RNA can do both catalysis and information storage, how does this resolve our conundrum? So, the way it resolves the conundrum is through what is known as the RNA world hypothesis, something which was proposed by Alexander Rich in 1962. So, what is this hypothesis? It is a multi-stage thing. So, the hypothesis says that right at the beginning, it is the RNA molecules which form and replicate. Now, these RNA molecules, some of them, not all, some of them develop catalytic abilities. These are the ribozymes, and the most successful ribozymes, they facilitate their own replication and catalyze simple metabolic reactions. So, they do both the job of the protein and the DNA, and this stage, this is what is known as the RNA world, where RNA is the primary molecule. This eventually leads to the development of proteins and DNA, which are more efficient for their own specific tasks. Why so? Because, okay, if you have ever worked on the bench and if you have ever tried to isolate RNA for any gene expression work, you will realize that RNA is notoriously unstable. Okay, so it is, well, I shouldn't say notoriously, because we are able to work with it, but it is pretty unstable, much more unstable than DNA, for example. Okay, and second thing is, if you look at how diverse an RNA sequence can be, okay, there are just 4 bases, right? Whereas if you look at a protein, there are 20 amino acids, right? So, for the same length, the amount of diversity in structure and therefore the amount of diversity in function that you can get in protein is way greater than that of RNA. So, because of this, by its very structure, protein is much more well suited for doing catalysis and all metabolic functions. DNA is much more well suited due to its stability for transfer of information across generations. And therefore, at some point, although RNA initially was doing both, at some point a subfunctionalization happens, because of which these things, these functions become specialized between these 2 molecules, DNA and protein. Now, there are several lines of evidence which are consistent with this view of RNA world. However, I will be the first to admit that not all questions have been appropriately ironed out. There are quite a few things that we do not know. So, for example, sure, I mean, it is a hypothesis that we went from RNA world to DNA plus protein world, but how exactly did the transition happen? Okay, what caused the transition? We can see how it will benefit, but how exactly was the trajectory? We really do not know. The second thing is, yeah, sure, RNA can do catalysis. Yes, sure, RNA can store information, but how good was it? Was it good enough for the whole thing to become sustained and well spread? We do not know. So, for example, for the longest time it was known that although RNA can help in replication, it did not really do a good job of it. It was definitely not as good as, say, DNA polymerase. However, very recently, 2024, last year, scientists have actually created a ribozyme which can very faithfully replicate another ribozyme which itself is a DNA cleaving molecule. Okay. So, you have one RNA molecule which is acting like a DNA polymerase and is able to replicate another RNA molecule which is acting like a protein. Okay. But chemically both are RNA, right. So, this is supposed to be one of the big breakthroughs in this field, and by no means this is the only breakthrough. There are several breakthroughs that keep on happening in this field simply because it is a very happening field, and so lots of work is going on in this, exciting new findings all the time. Okay. So, monomers can be formed, polymerization can happen. After polymerization, from polymerization to function, if you go through RNA, and then at some point hopefully RNA world gave rise to the current DNA and protein world. However, we still have a problem. What is it? Which is that if you need to have a system, it will be, you need the system to have some kind of a way to separate itself from the exterior. So, if you look at our present living world, barring the viruses which are anyway somewhere in the boundary, everything is enclosed in a membrane. Okay, everything is membrane bound, and this being enclosed in a membrane is actually extremely beneficial. Why? Because this enclosure allows for the creation of a unique internal chemical environment. Why is that important? Remember, I told you that initially when the molecules are being formed outside, then they are going to be unstable simply because they are going to react with the gases and pH and everything else in the ocean, in the open ocean. Whereas if you can create an enclosure, then that raises the possibility of having a different pH, a very different oxidation or reduction environment inside, and so on. So, that essentially increases the ability of the system to do a lot more if you can have your own unique chemical environment, and that is something that until and unless you have some kind of an enclosure is not going to happen. The second thing is, many of these macromolecules, for example, I told you about RNA, they are pretty fragile. So, if you are able to create that enclosure, you will be in a much better position to protect those fragile macromolecules, and that is very important if you want a stable inheritance to happen. I mean, if your DNA is simply fragmenting before it can have a chance to reproduce, then there isn't much life, right? And thirdly, the formation of these kind of enclosures can locally increase the concentration of the reactants. So, earlier we were talking about hydrothermal vents or clays and so on and so forth. If you are able to put things inside a boundary, then you can keep on increasing the concentration over there and have a locally high concentration of all these reactants. So, and as I said, we already know that all living forms minus viruses are membrane bound. So, how exactly could such enclosures or compartments form? Now, Oparin, he proposed that if you have organic molecules, they can form microscopic spheres which he called as coacervates, and these coacervates could then act as protocells. Okay. Nothing wrong with that. People have shown that coacervates can and indeed do form under many situations. However, there is another way in which such kind of membrane bound spheres can form, and that is through compartmentalization through what are known as amphiphilic molecules. Now, what are amphiphilic molecules? So, there are many molecules, the biggest example being fatty acids, which tend to have two parts in the molecule. There is typically a hydrophilic head, which means part of the molecule which likes water, wants to be closer to water or orients itself toward water. And then there is another part which is typically called a tail, which is hydrophobic, which wants to go away from water. Now, this is the nature of the molecule. Okay, there is no willfulness or free will or anything over here. This is the nature of the molecule. And as I said, the biggest example of this are the fatty acids, which can combine with many other molecules to form the lipids, right. So, we already know that lipids are the primary component or one of the major components for almost all the membrane bound organelles. So, turns out that if you put these fatty acids or even the lipids into water, then they can form different kinds of structures. So, the simplest kind of structure that gets formed are these bilayered sheets. Now, why is a bilayered formed? Because remember, these are the heads, these are hydrophilic, these are the tails, these are hydrophobic. So, if they arrange themselves in a bilayer, then the heads on both sides, okay, this part over here and this part over here, they are facing outside, they are facing the water. Whereas, the tails, the hydrophobic parts, they are facing inside, they are facing away from water. So, as a result, this is a somewhat stable structure, and people have actually shown that if you have proteins incorporated in them, then this becomes even more stable. So, now if you have a bilayer structure like this, then this bilayer sheet itself then can form different kinds of structures. So, the simplest structure that it can form is a sphere, a single layer sphere, which is known as a micelle. The other thing that it can form, this bilayered thing entirely can just go inside, and have a cavity inside, and that is what is known as a vesicle or a liposome. Okay. Now, the main difference between liposome and micelle is that this is single layered. Okay. So, you can see that you have the heads and you have the tails, and that is it. So, there is hardly, this cavity is very small, but in this particular case, this entire bilayer folds on itself to become the sphere, because of which there is, in this internal part you can have a large cavity, and once you have that cavity, then in that cavity you can have your proteins and you can have your nucleic acids and you can have all your cell organelles, etc., which can form later. So, the basic point is that you do not have to do anything, you just have fatty acids, lipids coming, and the moment you put them in water, these structures form. And more importantly, particularly these liposomes or the vesicles, they can grow by incorporating more fatty acids into the membrane. So, this is not a static hard sphere, this is something which can potentially keep on increasing its volume by incorporating more molecules, and obviously that is a very, very important thing both in terms of growth and in terms of forming another structure. So, if you have a large enough liposome, at some point it can simply bud off like this and become two liposomes. So, the structure or the flexibility of the membrane allows it to do that. So, this is why it is a very crucial step, the formation of lipids. So, you have these things, these liposomes, and hopefully inside these you have various proteins and you have various nucleic acids and so on and so forth. So, that is what we call a protocell. It is not a cell, it is a protocell, it is going towards a cell. Okay. Now, from the protocell, so these are the first lifelike organisms, the protocells, and from that protocell, we somehow need to come towards the so called last universal common ancestor. Why? Because I told you that all existing life forms except viruses, they are cellular. So, there are several lines of evidence which suggest that all the existing cellular forms have arisen from a single common ancestral population, and this population had certain properties. What are the properties? Okay, we will talk about that in a minute, but this single ancestral form, this is what is known as the last universal common ancestor, and this is, as I said, this is the hypothesized common ancestral cell population which gave rise to the three existing domains of life, the bacteria, archaea and eukarya or the eukaryotes, and this LUCA, our evidences, DNA sequencing evidences indicate, arose about 3.5 to 4.3 billion years ago. So, this is just to show you the whole thing in the context of the tree of life. So, this is bacteria, this is archaea, this is eukarya, eukaryotes, okay, animals, plants, fungi, etc., etc., and all of these finally come from one root node, and that root node is the LUCA. Okay. Now, there are a few points to be kept in mind over here. It is a hypothesized entity based on biochemical evidences. Okay, very important. We do not have a fossil form to which we can point and say this is LUCA. Okay, it is coming from hypothesis. It is a hypothesized entity, but based on very strong biochemical evidences, as we will see in a moment. Most importantly, LUCA is not the first life form. Okay. There most probably were other life forms that originated. I mean, we are talking about protocells, right? Different kinds of protocells would have originated. Different kinds of protocells might have had different properties. They might have had different kinds of combinations of proteins and nucleic acids. However, it is from LUCA, that population, that all the life that exists today has arisen, or so we believe. Why do we believe so? Because if you look at the genetic code, okay, that is almost universal with very minor exceptions across all life forms on this planet, right. So, we believe that this is because the genetic code comes from LUCA. Secondly, if you look at the amino acids, so you know that amino acids can come in two different kinds of stereoisomers, the so called L forms and the so called D forms. Now, turns out almost without exception, all life forms on earth, they only use L amino acids, nobody uses D amino acids, okay, or very few. However, when it comes to sugars, we all use the D sugars, barring very few exceptions, nobody uses L sugars. Okay. Similarly, there are a core set of biochemical pathways which are common across all life forms, from the lowliest of the bacteria to the mightiest of the elephant. Why exactly is this happening? Again, as we pointed out, when we were looking at the evidences for evolution, that the simplest way to think about it is that this is because of a LUCA, plus universal common ancestor. And similarly, if you look at the machinery of the cell, the ribosome, the way the ATP is synthesized, okay, many of these components are universally conserved across all life forms. So, what were the properties of LUCA? Now, before you ask the question, the first thing that you ask is, how do you even figure out this thing? Okay. So, the simplest way to figure out is by comparing the common genes and properties of bacteria and archaea. Now, why are we saying bacteria and archaea? Why are we excluding eukaryotes? That is because we have very good evidence, as we are going to discuss, that eukaryotes arose from bacteria, a combination of bacteria and archaea. Okay. So, the two initial domains are bacteria and archaea. Now, if you try to look at genes that are common across all organisms that we have sequenced till date, there are about 30-35 such genes, and those are mostly related to ribosomes. Okay. But that does not give us too much of distinguishing power. So, what people have done is they have taken the known bacterial and archaeal sequences, and under various kinds of assumptions they have tried to figure out what are the common genes and the common properties between these two groups. Now, of course, it depends on what assumption you are making. So, this is a very nice paper, this particular 2018 paper, where they have made the various assumptions and they have shown what are the consequences of doing that, taking that assumption and not taking that assumption. So, anyway, long story short, after doing all this stuff, what people have generally agreed about the common properties of LUCA are as follows. A, it was single celled, it was lipid bilayer membrane, it had lipid bilayer membrane and the cytoplasm was water based. It used DNA as its primary genetic material, and it could replicate the DNA and transcribe it into RNA. It already contained genes, it already contained the universal genetic code, and it possessed ribosomes to translate RNA into proteins, had a core set of enzymes for DNA repair and genetic recombination, and it multiplied by duplicating its contents and undergoing cell division. So, these four sets that I am talking about, these are almost universal across all living organisms, which is why we are very sure that they are most probably very good candidates for being LUCA properties. The next set I am going to give you, they are mostly based on coming from this particular paper, based on comparisons between bacteria and archaea under various levels of assumptions. But from what we understand, it looks like LUCA was a chemoautotroph. It produced its own nutrients using inorganic chemical reactions, and looking at the genes that seem to be common across the bacteria archaea, looks like it reduced carbon dioxide and oxidized hydrogen and used acetyl thioester as a key intermediate in its metabolic pathways. So, if you are a biochemist, this should tell you that there which kind of biochemical pathways are used and how they differ from, for example, what happens in eukaryotes today. And mostly it looks like that it was anaerobic, it was a thermophilic form, and therefore most probably it lived in high temperature environments. Could it be anaerobic hydrothermal vents? Maybe, very likely candidate. Do we know for sure? No, we cannot know for sure, but looks like that is where you would find a LUCA. So, the main point that I want you to take home from here is that LUCA was not a simple protocell with a few proteins and a few nucleotides thrown inside it. LUCA was already a metabolically highly sophisticated organism, which was, it looks like, already adapted to an extreme environment, right? So, this means that we have a gap in our knowledge. What is the gap? When we were talking about the Oparin-Haldane hypothesis and the various steps of it during this discussion, we went up to the formation of protocell, and then we came from the other side and we talked about LUCA. We don't really know how we went from the protocell to LUCA. Okay, so that connection, what are the things that led a simple bag of liposome containing some macromolecules to something as sophisticated as this? We really have no clue. But we definitely know that it's very, very, very unlikely that the initial protocells were as complex as LUCA. So, as I said, we don't really have any answers, but you can bet your life scientists are working really, really hard to figure this out. Now, at this point, I would like to quickly recommend a video to you. So, we covered abiogenesis in quite a bit of detail, but this particular video by Arvind Ash, this mostly talks about the concepts that we discussed over here, takes a more lipid first kind of a view, but it also mentions a few papers that we didn't really cover during our discussion, has very nice animations. So, it is highly recommended, and the components of this are part of the course. Now, we need to talk about the concept of major evolutionary transitions. What are these? So, major evolutionary transitions are fundamental shifts in how living things are organized, and they typically happen when simpler independent units join together to create a new, more complex, but in a unit which has many interdependent parts. Okay. So, remember, when we in our first discussion, we were thinking about what are the various properties of life. We said that one of the things that we see is that most living forms, even the simplest bacteria, are pretty complex, and there are lots of components, and these components seem to be interacting with each other, and you have a hierarchy. So, you have single celled organisms, then you have simple multicellular organisms, then you have more complex multicellular organisms, and so on and so forth. And as you go up the hierarchy of complexity, the level of complexity, how many interdependent units, all that keeps on increasing. So, major evolutionary transitions refer to those situations where you go from a relatively simple level to a more complicated level. And these transitions, as I am pointing out, they often lead to increased complexity, and the fundamental change that happens is how information is inherited and transmitted over time. In other words, how selection ends up operating, not the basic mechanism, but what is the unit of selection, that is what typically ends up changing during these major evolutionary transitions. And more often than not, some kind of a cooperation across multiple entities. Okay. So, for example, different kinds of cells cooperating to form a tissue, different kinds of tissues cooperating to form organs. So, these are what I mean by cooperation across multiple entities. Okay. Or for example, honeybees cooperating with each other to forage for food. Those are the kind of cooperations that we are talking about that often ends up becoming a crucial factor. If this is not making too much sense, just wait for a minute, I will give you the major transitions that have happened. So, whatever we discussed till this point, about how you go from simple, lots of polymers to a proper bag containing these polymers, this part, this sequestration of majority of transmissible information in the genes, and then the formation of cell membrane to create cellular life form. This is typically cited as the first of the 6 major transitions in evolution. The second major transition that happened was the formation of eukaryotes. Now, why am I singling out eukaryotes over here? Because remember, all the multicellular organisms that we have, all of them are eukaryotes, including us, obviously, right? So, turns out that the formation of eukaryotes, also known as eukaryogenesis, obviously, is a very, very interesting and a special event. Why? Because the theory goes that, well, it's no longer a theory, we more or less have extremely good evidence to indicate that this is what happened, is because it happened through a process known as endosymbiosis. So, what really happened? So, if you remember, I told you that you had LUCA, and then from LUCA you had 2 major domains, the bacteria and the archaea. Now, turns out that at some point, some anaerobic archaea bacteria that ended up engulfing a an anaerobic archaea, it ended up engulfing a bacteria, an aerobic bacteria of type Proteobacteria, about 2.2 billion years ago, and this merger became very, very important. Okay, this merger is what led towards the formation of eukaryotes, and these aerobic bacteria that were engulfed, that were incorporated, these were the ones that formed the mitochondria. And turns out that this was about 2.2 billion years ago, and then at some point about 1.6 billion years ago, there was one more such event that happened, and in this particular case, an aerobic eukaryote which already had the mitochondria, there was one more incorporation of a bacteria, but this time it was a cyanobacteria, and this cyanobacteria now ended up forming chloroplasts, and this lineage is what led to the formation of the green plants. Now, the formation of green plants is very, very important, because remember, they are the ones which are fixing carbon dioxide and giving rise to oxygen, which is very important for the oxidating atmosphere that we have around us today. So, these 2 endosymbiotic events is what leads to the formation of the eukaryotes as we know them today. So, just to show you the major transitions. So, here is the first transition that we talked about, separate genes and cell membranes came together to form genomes within the cell. So, we had compartmentalized genomes, and this is what allowed the formation of large complex genomes and everything else associated with them. At some point, separate unicellular organisms, they ended up forming symbiotic relationships with the incorporation of the mitochondria and the plastids. So, that is how eukaryotic cells became formed, and once eukaryotic cells became formed, then there were transfer of genes between the mitochondria, plastids and nucleus and vice versa. So, that is how the genomes themselves inside the organism became hybrid from multiple sources. So, the remaining major transitions, at some point asexual unicellular organisms became sexual unicellular organisms. Okay, so, sex evolved, and this is where meiosis ended up coming, and we have already discussed how sexual reproduction and in what ways it could have speeded up evolution. Then at some point unicellular organisms became multicellular organisms. The point, that is when you could have differentiation at the levels of cells and the differentiation at the level of tissue, and then there was the germ soma separation that ended up happening. We have discussed the importance of all these things. Then at some point the multicellular organisms, they led to the formation of eusocial societies, the bees, the wasps, the ants that we discussed. Of course, this did not happen across the board, it only happened in a few lineages, but wherever it happened, the reproductive and the non-reproductive functions got separated out in two different organisms, okay, which is a huge thing. Otherwise, barring these eusocial organisms, in all the cases the same organism has to function and perform both functions, both reproduction and everything else related to body maintenance. It is only in multicellular organisms that you have these two functions getting separated out in two different bodies. So, which is why many people have even argued that the entire colony of an ant or the entire hive of the honeybee is an organism. That is a different philosophical argument. And finally, different species, they came together to form mutualistic associations, for example lichens, okay, and that is when you had physically, in evolution of physically conjoined partners, various kinds of endosymbionts, and that is supposed to be another very major transition. So, this entire idea of the major transitions, this comes from this famous book by Maynard Smith and Eros Satmari, The Major Transition in Evolution, and generally this is supposed to be one of the major landmark books in evolutionary thought. Okay, so till this point, one thing that we did not cover was what really happened after the eukaryotes came in. And of course, a lot of evolutionary biology, macroevolutionary biology is there, which group led to which group and so on and so forth. Unfortunately, we do not have time to cover that bit, but I will make two recommendations, two videos. They are not a part of this course, they are more for your own information so that you have a greater understanding of the whole topic. So, the first is this 36-minute video called A Timeline of Life on Earth, 4 billion years of history, by this channel called SciShow. Very fast paced, very informative, in fact lots of information actually, and overall pretty enjoyable, nicely done. Okay, they use simple photographs, they do not have animations or they do not have very fancy photography, but nice stuff. The second one is The Evolution of Life with David Attenborough. It is 48 minutes or so long, it is slow, but it is majestic. The photography is mind-blowingly superb, as you can expect from a BBC documentary, and of course you have Sir David Attenborough in it. Okay. So, between these two you should get a lot of information about the other macroevolutionary changes that happened in the various groups. So, now that we have looked at the evolution of the other life forms, we will turn towards the one species that interests us the most, which is of course us, right. So, we are now going to look at what or how exactly did human beings evolve, are they still evolving, and so on. But that is what is going to happen in our next discussion. See you then, bye.